All right, guys, let's go ahead and get started here. I know you guys are itching to get out of here because you just had an exam. Hopefully it wasn't terrible. Okay, that sounded positive. Not that I can blame you guys. End of the semester, you don't want to be taking more exams. I have unfortunate news for you that next week's a little hard for lab, but I personally think it's very fun. But there's at least one part that I think most people find very fun, which is we'll be making yogurt in lab, which most people enjoy. So we'll talk a little bit about that. So we're going to be basically doing two labs kind of combined into one. And the reason why is there's a lot of kind of wait and sit around and wait in between certain things. So that's why we're kind of putting two labs together. So we're going to be doing food safety. Food safety can kind of be summed up in two things. But smelly foods can be okay. So we can actually have a certain amount of bacteria in certain foods. And sometimes it's beneficial to have certain foods be fermented, for example. And fermented foods actually have really great gut health benefit. But sometimes there is a bad point in which bacteria are in our foods. And once we kind of reach that bad point, or especially if it's a certain type of bacteria, there's no safe limit, that can be a very crucial issue. And so we do have to keep an eye out for that. Oops, excuse me. There are a couple of different ways we can test for this. One of them is we've actually done already in class, which was viable plate count testing. Another one is the water testing we did. Those are both examples of what we call quantitative analysis. But realistically, this type of test, which is SNS Auger, is a qualitative test. We're basically checking, hey, is this type of bacteria or bacteria groups present? And if it is, that's a problem, and we should do further testing if we see a positive from here. So it's just looking for the presence. It's not necessarily trying to tell you the amount there is. So the way this test works is it is looking for, as the name implies, Salmonella and Shigella. It is a very good primary food safety auger plate. There's a couple different versions that look for Salmonella and Shigella. This is the one that we're using. It's pretty simple. What it looks for is it looks for the ability to ferment, which is actually counterindicative of Salmonella and Shigella because those two organisms do not ferment lactose. And it looks for the production of hydrogen sulfide, and Salmonella does produce hydrogen sulfide when it grows on this plate. So it's actually really interesting. The way it works is it's selective for gram negatives. It's actually very kind of stringent about the number of gram negatives that can grow on here. It doesn't just allow any gram negatives, so it's actually kind of really bizarre. It tends to kind of heavily favor Salmonella and Shigella. But there are some other organisms that do grow on here. So we're going to talk about that. It is selective for gram negatives. It does a selectivity through bile salts, a chemical called brilliant green, and a variety of citrates. And what these do is they basically prevent gram positives from growing in here at all, and it kind of inhibits and messes with the growth of some gram negatives, which is also a reason why this media is not super amazing for its intended purpose, because it will actually inhibit some Shigella species from growing on here, which isn't very handy. But there are some other bacteria that can grow on here, including things like E. coli. So this is an example of an SNS plate. SNS plate kind of has this little weird red coloration to it. It's kind of like an off-colored plate. And E. coli on here will create a pink coloration to the bacteria, much like MacConkey's. Not exactly identical, but very similar. And it'll create that kind of pink coloration if it's a lactose fermenter. But Salmonella and Shigella are not lactose fermenters, so they will never show up as pink in this. Shigella, if it grows on here, will create nice clear colonies that will be able to survive on this plate. And Salmonella, if it grows on here, will create this hydrogen sulfide. If it produces a hydrogen sulfide from something called ferric citrate, which is one of the citrates in here, but it will create a kind of this, what we call a fish eye colony, where it's got a very kind of clear jelly-like colony, with the center of it having a dark black spot from hydrogen sulfide causing ferric citrate to precipitate. So it's very, very similar to the way the TSI slants we used were. A couple of different chemicals here and there. but basically we're looking for that precipitation and that black spot being created. And so we want to look for that, and that would be a positive indicator of salmonella. So what we will give to you guys next week is we will give you guys a little tube of meat that may or may not have some bacteria in it. You guys will take a swab that's sterile. Each person in your group of 4 will get a plate. You'll swab the meat and you'll streak it back and forth on this auger. And then you guys will throw that swab in a bucket of bleach. Really, really important part here. This auger is it's softer than you guys are used to, so much softer than TSA or Mueller-Hinton auger. So you can see I was a little rough with this guy, and what ended up happening is the plate ended up cracking because I was a little too rough with it. So be very gentle as you guys streak back and forth on here, okay? Don't be too rough with it. It's a little bit softer than you'd expect. Once you guys are done streaking with it, and you dispose of the swab, you then are going to take the plate and you are going to seal it with parafilm. And the reason why is the bacteria that are probably on your sample, or maybe not at all, some of them are going to be, because we're still using fresh ground meat, it could actually have a potential pathogen present on that meat. We don't know for sure. Even though it's unlikely, highly unlikely, we don't know for sure. So we take extra precautions. Even though this media can inhibit the production of toxin-producing bacteria or the toxins that those bacteria produce, it doesn't necessarily guarantee it. And so for an extra layer of protection, we will seal these with parafilm so that they're completely sealed, and then we will tape them shut and put them in the incubator upside down so they can grow. So that's kind of an important, unusual step to what we normally do. We're going to seal it with parafilm. Okay. And then we'll incubate them and check to see the results. Now, because you guys will all have the same tube from the group of 4, keep in mind that this is going to be a little bit more, you might have some people that, your group might all have the same results, so you might all be negative, or might all be positive, or might all have E. coli present. So everybody in the lab section is going to kind of have to share their plate so that everybody can see what a positive and a negative or a somewhat negative result looks like. Okay? All right. We will also be we're not going to be making sauerkraut because that's a huge hassle. Instead, we're going to be making yogurt. Yogurt is awesome. However, I must stress highly, these are forbidden lab foods. We do not eat them. You don't put any yogurt that we make in lab in your mouth. The yogurt that is already present in lab to do this experiment with, do not put in your mouth. The milk that is in the lab, do not put it in your mouth. Also, the meat tube, I shouldn't have to say, but I'm going to, this should not put in your mouth. Nothing in the lab we ever put in our mouth. Even though it is technically food and it's theoretically safe to consume, we're not going to because we assume it's contaminated because it's in a BSL-2 lab. Okay, great. Everybody clear on that? Yes? All right. I only say this because, somebody might forget. Okay. What you guys are going to do here. Yogurt is basically just a milk product that's been exposed to 2 specific genuses, and each of these genuses have different bacteria within it and subspecies within those, called Lactobacillus and Streptococcus. And there are specific ones that basically make this guy thicken up and create nice creamy yogurt from milk. We can actually steal yogurt that's already been made, that is basically a starter culture with these in them, and that's exactly what we're going to do. To start this process, we're going to take whole milk and we're going to re-pasteurize it. So normally the milk you guys buy from the grocery store has already been pasteurized. We're going to re-pasteurize it because we don't know what levels of bacteria in that milk, because pasteurization does not kill all the bacteria, it just lowers their numbers. And since it's sitting on the shelf, maybe its numbers have gone up slowly, even though it's been chilled out in the refrigerated section. So we're going to reheat it up to pasteurize it. And the way we do this for our specific purposes is we are going to take a sample of this and heat it up to 72 degrees Celsius. And once it hits that, we really don't have to keep it for like 2 seconds there. We're going to cool it off in an ice bath and let it chill out and go down. And once it goes down to a nice, normal, like touchable temperature, we'll then put it in a small cup, styrofoam cup, 4-ounce cup. And then we will take a spoonful of yogurt, a tiny spoonful. You don't have to do a big glob, just like a little spoonful. And you stir it in there, mix it in so it's nice and easily dissolved. We'll then take a layer of saran wrap, stick it on there so gets all the air out. There's no air present. And you will then cover the entire thing, the top of it, with aluminum foil, and you'll wait 24 or 48 hours. And after you take it out, you should have yogurt. Now, the reason why we use the saran wrap is because we want to get out as much air as possible. The bacteria that can utilize air, aerobic bacteria, tend to actually just go after the lipids and cause that spoilage smell really strongly. The real big primary driver of this is one of the most common bacteria in all of nature, which is Pseudomonas aeruginosa. I think Pseudomonas aeruginosa actually represents a fourth of all bacteria in the world. That's a huge number. So it's pretty common. So we want to try to minimize its ability to grow and thrive. So that's why we do that. And then we want to protect those bacteria by covering them so they're not exposed to sunlight. It tends to cause a lot of other problems too. So we just want to keep it covered. While these two bacteria are in there, they are going to be munching away on the lactose there and fermenting it. And as it ferments, it's going to uncoil that DNA, allow it to thicken up and become more congealed, and that will make our yogurt. That's also what makes yogurt kind of slightly tangy, is that fermentation process and the byproducts therein. All right. Does anybody have any questions on that part? All right. This week, you guys are lucky we have 2 science things of the week, so we'll go over that. This one's actually really easy, I promise. Okay, that's a lie. It's really complicated, but I'm going to make it easy. It is actually really easy. Direct activation of bacterial innate immune system by viral capsid proteins. Did you guys know that bacteria have an innate immune system? So humans, we have an adaptive and an innate immune system. Our adaptive immune system learns from infections and develops its own specific, unique way of dealing with them. And our innate immune system is looking for built-in properties of bacteria that are hard for bacteria or viruses to change, that we can quickly and easily attack if we see them, so kind of like a tripwire alarm. And then we can come in and handle them quickly and effectively. A lot of people actually make the mistake of saying, oh, our innate immune system isn't as good as our adaptive immune system. Actually, they're very, very important to one another, and the innate immune system has a very big starter effect and continuation effect for your adaptive immune system. We do know of instances of people with parts of their innate immune system that are missing, so certain cell lines or cell types that are missing, and those people have severe health problems throughout their entire life. It's actually quite detrimental not to have those innate defense mechanisms in place. But one of the things that we've been learning more and more about is that bacteria have an innate system too, which you wouldn't think of, but they actually do. And this one's actually really cool. So a phage will come in, it will shoot into a bacterial cell, and it'll start going away and making copies of itself and the little proteins it needs to make the more of its capsid, this guy right here, the major capsid protein. And bacteria can produce a type of gene that's a weird little combo gene called, it's got a synthetase part, and goodness, what was the other one called? It is an antitoxin part. The antitoxin part is kind of wacky because it is attached to the synthetase, like that, but it's its own little kind of discrete region, and it binds to the synthetase and inactivates it. But that antitoxin part also has another little part to it that's looking to bind major capsid proteins. And if it binds major capsid proteins, it unlatches from the synthetase, and once it unlatches from the synthetase, it starts making, it basically does a series of things that cause your body to stop being able to make protein, not your body, causes the cell to stop being able to make protein effectively, and basically causes that bacterial cell to kill itself. And you might be thinking, well, that's kind of not very helpful, but it's actually kind of what our own cells do. If you get infected with a viral cell, or a viral particle, and your cell gets infected, your immune system will come in and say, hey, cell, you gotta die because you're infected, so you don't kill the rest of us. Same thing here. This bacteria will go, oh, I'm infected with a phage. I should die off so I don't make more phage copies and infect the cells around me. And it is an incredibly effective system. So this is the example right here. I know I'm gonna make it a little bit larger because it's hard to see. Hopefully that's easy. All right. Right here, these are plates with bacterial lawns in them, and these little clear zones here are the plaques where phages have been introduced at different concentrations. And you can see that right here, the lower concentration, the phage, basically 0, The phage doesn't do anything, but as you start adding more and more phage in, it just starts eating a hole where all the bacteria are, and it kind of kills off all the bacteria. Jeez. Images, obnoxious. But, okay, let's just delete you. Make it easy on myself. But if you have that capsid protein sensing bit, if it's there, it will start killing off the cells that are infected by the phage, and the phage has to be at really high concentration to actually start making a plaque. So you can see over here, it starts making a plaque just at this very minimal thing, but it doesn't actually kill any cells over here. Because those cells die off that are infected by the phage so quickly, the other bacteria are able to move into that area even though those cells are dead, because the phage isn't being produced. All the phage is dead because it can't survive on its own. So that's actually really, really cool. So there you guys go. Bacteria have an innate immune system. Fun. All right, does anybody have any questions on the food lab or the cool science thing we just talked about? Make sense? Okay. We're going to now talk about PCR and gel stuff, which is the wave of the really old technology that's still used today. It's actually been around for a really long time, and we've been more and more efficient at it so that it is incredibly fast right now. We are able to do some crazy things that we weren't able to do even 10 years ago, but even more so than when we first started doing PCR, which I'll talk about in a second. Okay, so PCR. Does anybody know what PCR stands for? Okay, PCR stands for polymerase chain reaction. And you will probably hear me at some point in time today go PCR reaction, which is the same thing as ATM machine, and it's dumb, and I shouldn't do it, but I still do it because that's the way I first learned it. Polymerase chain reaction. So we're using DNA polymerase. We're doing it in a series to create a chain reaction to make a new strand. That's all that's happening. You basically only need 4 components to do this, which is template DNA, primer DNA, nucleotides, free nucleotides, and polymerase, which is not shown in this picture, but you just assume that it's there. And there are 3 basic steps to any PCR. The first step is denaturation. Denaturation is where you get this really high temperature and it causes your template DNA to pull apart. This has to be the highest temperature because template DNA, if we just assume it's maybe genomic DNA, is really big and it doesn't want to separate. It wants to stay together. So you have to heat it up a lot so it pulls all the way apart. Once it's all the way apart, we can lower the temperature, but not all the way, down to about, so this temperature is about 95 degrees Celsius. We're going to lower it to about around 50 to 65 degrees Celsius. It depends on your primer. And we call this the annealing step. And this will allow your primer to bind onto this little region of single-stranded template DNA over here, and this little primer to bind onto this sequence of DNA over here. And now it's stuck because the melting temperature of primers, the temperature at which primers stick together and pull apart, is a lot different than the template DNA. So they can actually stick together a lot easier. So they aren't pulled apart as, they're not as affected by the high temperatures. It's actually really handy. Okay, then when this happens, we will then raise the temperature just a little bit to about 72 to 75 degrees Celsius, which allows the polymerase to come on and then start the reaction making new strand of DNA. So it'll latch onto the primer and template together, and then it'll start going along here and making new little copy of DNA. And then we repeat this process. We melt that all apart. We add in more primers and let those primers come together. And then we put in more polymerase to come in and make another copy, and so on and so forth. We repeat this a bunch of times, usually around 25 to 40 times. The number of cycles is just dependent on what you need to do for your particular process. And this whole cycle is actually really, really interesting because we have an automated machine to do this now. Back in the day, they used to have a poor little graduate student sit there with beakers of hot water set to specific temperatures, and they would take one test tube and move it from beaker to beaker with little timers going for every time they had to move it. And every time they had to move it, they had more reagents because the temperature here, 95 degrees Celsius, is actually really super ultra mega high destructive to DNA polymerase. Our DNA polymerase cannot stand that. It has to be a nice, body temperature to work, which is inconvenient because DNA has to be pulled together. So if we're not using what we normally use in our cells, which are different types of gyrases and isomerases and such, we need something else, and we use heat. Now I'm going to explain how we get around this. The way we do this PCR, we use something called a master mix, which has all the chemicals required. So I said that there were like 4 things. It was kind of a lie. There's a bunch of stuff that you need for it, but 4 major components that actually do something that is relevant. The first thing you need is DNA polymerase, and we actually use a very important type of polymerase from a very specific organism. We call it Taq polymerase. Does anybody know which organism it comes from? Does anybody have a wild guess? I'll give you a hint. It's an organism that does really well in heat. Okay, so this one's kind of trippy. We decided to steal the DNA polymerase from an organism found in the geothermic vents, so geysers, in, I believe it's Yellowstone National Park that has the big one. Yeah, we stole it from that bacteria. So there's this bacteria called Thermophilus aquilus. I think it's called Thermus aquaticus now. And we just stole it from them because we were like, hey, there's organisms living in this like near boiling water. How does it do that? Well, it turns out it does it by having specially designed polymerase that can handle super high temperatures. So we've done this, we've stolen it, we've modified it a whole bunch, and now it can survive even higher temperatures. It's actually really cool. We also need buffer to keep the reaction happening at a good pH. Inside, we also need these nucleotides. These are the nucleotide triphosphates that you need to use to make a copy of DNA. Magnesium chloride and calcium chloride. Magnesium chloride and potassium chloride, because those help stabilize it binding as well as accuracy of the polymerase reaction. And then a little bit of ammonia sulfate and some Triton X. Triton X is an anti-bubbling agent, which is very, very handy because bubbles are really bad for this reaction, and I'll explain in just a second. And usually most of these master mixes you can buy now instead of having to make by hand, and you can buy them and they have some enhancers and stabilizers in them. The last things that you need to do or add are things that you need to add yourself every single time, which are primers, in this case a forward primer and a reverse primer, which we will actually do for you guys. And the last bit that you guys will do is the template, whatever the template is. In this case, we will be using our unknown bacteria as our template. We will give this whole mixture to you guys, this 20 microliters, pre-made for you guys. Because I don't trust you. No, that's actually not true. It's because that's a huge pain in the neck to pipette yourself, and especially 20 microliters is a little bit difficult for people to do. So I just do it for you guys. So you guys don't actually have to worry about that part. Part of it is also that it can be a really terrible pain if something accidentally happens. And I'll tell you guys a terrible story of what happened when I was an undergraduate student. Back in the day when we had to make the master mixes ourselves by hand, we had a little tiny tube of this stuff called Taq Gold, which was super duper expensive. It was in a vial about this big. It was 100 microliters of liquid, and it cost about $360. And I spilled an entire tube of it once. That was not a fun day to go talk to that professor because that was a lot of that polymerase. You only actually need you only need a quarter of 1 microliter to do a reaction, and so that was a big issue that I messed up. So hopefully you guys never have to experience that because that's not a fun conversation. Okay. So what we'll do is we will give you these tubes for you guys, pre-made. For you guys, because you guys only have one organism, each person will get 1 PCR tube, and you guys will take your tube, you will take your slants that you guys have made, you will use a pipette tip and gently grab just a teeny tiny amount of bacteria. You don't need a bunch of bacteria here. Less is more, and I'll explain why in just a second. And you don't want to accidentally grab any agar along with the bacteria, just the bacteria on the tip of the pipette tip. You can actually put it on a pipetter to do this, and then you're going to put it into the tube with the 20 microliters and gently swirl it around. And you can also gently pipette up and down to help knock off some of the bacteria so it's evenly distributed in the PCR mixture. In this, you want to be very, very careful because in this up and down motion, we do not want bubbles. No bubbles. The reason why is because bubbles interfere with the heating and cooling, and because air is actually an insulator, and it will not allow things to get to the right temperature. So we do not want that to happen. So make sure you guys are very careful not to create bubbles. Once you guys get that done, even if you do have a little bit of a couple of bubbles, it's okay, because we will stick it on a centrifuge, take it up to your TA, they will spin it on the centrifuge, and then we will put it on the thermocycler, which is that machine there that actually does that automated thing that we used to have a poor graduate student do with beakers of water back in the day. It actually does all of these things automated for us in cycles, which is great. And it'll do it for us automatically. You'll put it in there, we'll turn it on, we'll let it run, and then the next day you guys will come back and we will do the gel part of this. So it's actually really, really handy. This guy is super fun to program and play around with, and there's a lot of cool things you can do with them now. One of the actual interesting ones, this was actually considered a luxury feature back in the day when I was an undergraduate student, is a heated lid. Because one of the things when you're dealing with reactions that are so tiny that you're bringing up to 95 degrees Celsius, 95 degrees Celsius is really close to the what? Boiling point of water. So your entire 20 microliters of reaction might vaporize, which is really bad because reactions don't happen in vapor. So what we do is we used to do back in the day is we used to add layers of mineral oil on top, which was horrifically obnoxious, especially when you get to get the oil out. But now what we do is we have heated lids, because to heat the lid higher than the temperature on the bottom, the liquid won't evaporate because the liquid likes to stay wherever it's coolest. And so if you heat the lid higher than the bottom temperature, then the liquid doesn't evaporate. It's super convenient. It is like magic. The oil one was terrible. Never do that. Okay, running a gel. How do you run the gel? Well, you guys will take a gel. It's actually got this dye embedded in it that binds the DNA. And you guys will actually take a turn making these for your classmates. So you will use a gel made by the previous lab section before you guys in doing your gel. So you will use their gel, and then you'll make the gel for the next section after you. So you guys will take a gel, you will put it in a gel box, and you will cover the gel box with an even amount of 1x TAE buffer. Buffer has some salts in it, so it conducts electricity, and you want to cover the gel about a centimeter or so above the gel with liquid. Once you do that, you guys will gently pipette in some of your reaction mixture. Now your reaction mixture is just kind of clear liquid, and that's actually really hard to pipette. So to help you with this, we will add something called 6x loading dye to your PCR reaction after it's all done. 6x loading dye does two things. One, you can see it, which is really convenient when you're trying to pipette something into something that's clear. If it's different color, then it's a lot easier. Two, it is thick with glycerol so that it sinks, which is very, very helpful when you're trying to load things in gel, because it wants to drop down straight down, and that makes it very easy for you to do what you need to do. The last thing it has is it has dyes in it that actually track along with DNA, so you can see as the dye is moving through the gel where your DNA can be. So you can see right here it starts, and if you look right here, you can see a little bit of separation of these dye bands, and so that you can actually guess how far in your gel your DNA has traveled, so you don't accidentally have your DNA run out of the bottom of your gel, which is bad. After you guys get them all loaded up, and real quickly I want to point out for you guys as well, lane 1 and lane 10 you guys will not use. That will be for your TA, and they will put in ladder here, and we'll show you guys what ladder is in just a second. But leave lane 1 and lane 10 alone. Don't use those ones. You will then close the lid, turn it on, and let it run. Now the most important part here is to be very gentle with your gels, because if they snap in half, we can't do anything about that. We have to throw them away. But the second part is we want to make sure that they are well side towards the black electrode. The black electrode is negative, the red one is positive. DNA runs from the negative electrode towards the positive one. If you have it the other way around, the DNA will run out of the top of your gel and then you'll have nothing. So once you put the lid on, you want to wait a little bit. And what you're going to do first is you're going to check to see that bubbles are running in the buffer tank. That means that the electricity current is running through here as bubbles are being generated. In this case, bubbles are good. That means it's running. And then you'll check over here in about 5 minutes, and you should see some of your dye penetrating this direction into your gel and having a little bit of that separation. That means everything is working as intended. Once it's done, and it'll take about 45 minutes to do, hopefully, we will then take your gel out and we will put it on a transilluminator, which will shine a light frequency that binds with DNA and that dye we added. The DNA and the dye that we've added will bind to each other and fluoresce when they're together, but nowhere else. And what you should see is something like this. And this is an example of one that I did in lab of a bunch of different organisms just last week. And you can see that we expect a band of about 1,500. This is our ladder. The ladder is known sizes. And so this band is 1,500. And you can see that we got bands from our PCR reactions of 1,500. Except for this guy right here and this guy right here, and I'm going to explain why. That middle band, I was a little careless because I was working with a bacteria that didn't have a lot of growth on it, and I was not trusting, and I dug a little too hard and grabbed a little bit of accidental agar with it. And agar has sulfates in it, which interfere with the PCR. That did not allow me to get a good band. This one, though, didn't have that issue. It was luxuriant growth. What I did is I grabbed too much bacteria. Some bacteria like to form a lot of biofilms. They're thick and viscous. You don't need a bunch of bacteria on here. And I grabbed too much, and that interfered with the PCR. And I almost said PCR reaction yet again. It's a hard habit to break out of. And that didn't do that. Didn't do a very good job. Now these bands are actually here if you squint really, really hard. So this is what it looks like in real life. There is a filter over this, but a physical filter, which is an orange filter. But if you squint really hard, you can see the bands right here. Sometimes it's easier for people's eyes to flip it to black and white, which is what I do oftentimes. But you can actually see right there, there's that little like band right there. That is a successful PCR reaction, but it's very weak. It didn't work very well. Now, while your gels are running, which take about 40 minutes or so to run, you guys will make gels for the next class. And the way you guys do it, you guys are going to make a 1% agarose gel. What you'll do, your TA will call you up one group at a time. You'll measure out 0.8 grams of agarose, add it to a flask with 80 mils of 1x TAE buffer. You will then microwave it until it's fully dissolved. Do not let it boil over. And there's a word of warning here. A, you need to give it a good swirl every now and then, but B, be very careful to watch it and not have it boil over, because if it boils over, it'll A, be very hot, and B, be very sticky, because agar kind of clumps up together and it'll make a giant mess and you have to clean it up and it's not fun. So just kind of keep an eye on it, make sure it doesn't get boiling over the top. It will boil a little bit. You just kind of have to keep heating it up until it's perfectly clear, like crystal clear. Once it's crystal clear, you will then take a moment, let it cool down just a slight amount till it's about kind of getting cool to the touch, and then you'll add about 8 microliters of this dye called either Easy Blue Light Dye or Gel Red, or what we will typically use in our lab is called Cyber Safe. You'll add about 8 microliters to that, swirl it around, and then you will pour it into a gel frame that we'll have set up for you guys, and add in a comb to add the holes that you put the DNA in after it's set. And you'll pour it in the frame. You want to make sure that there is no bubbles in the gel, because bubbles will actually cause DNA to bend around the bubble and it messes things up when there is. So you'll actually take a little pipette tip and like poke or pop out any bubbles that you have. Your TA will help you with that. Once that's done, you'll take the gels out and you'll wrap them in saran wrap with some buffer to kind of keep them moist for the next class. But then you guys are done. Real quick, agar and agarose are 2 different things. This is actually important because a lot of people forget this. Agar is actually a component of agarose. So agarose is like the crude extract, and agar is taken out. The other part is this stuff called agaropectin, which has sulfates in it, and we don't, excuse me, we don't want those in the gel because they don't let They don't make a very good clear resolution gel. Hold on. All right. All right. Any questions on that so far? Makes sense? Yes? Good? All right. Other cool science thing of the week. This one's actually really interesting little guy, even though it's got probably an incredibly complicated title, which is Whole Genome Sequencing Facilitates Patient-Specific Quantitative PCR-Based Minimal Residual Disease Monitoring in Acute Lymphoblastic Leukemia, Neuroblastoma, and Ewing Sarcoma. What does that mean? Okay, it's using whole genome sequencing, so we're taking cancer cells from patients, we're sequencing the entire cancer cell. We're getting a whole genome of it for that specific patient. So it's patient-specific, which is actually really cool. And then we're using that to test after they have treatment for their cancer. So be it radiation, surgery, chemotherapy, what have you. We're looking and using that as a test for teeny tiny amounts of it maybe coming back. So it's a monitoring system after treatment. And the reason why this is important is because if we can detect really tiny amounts of this starting to come back, we can actually treat it much faster using easier methods such as normal regular old chemotherapy or radiation, rather than finding out when it's a much larger mass and bigger problem. Why do we pick these three in particular? One, it's because some of these do not have very good markers that are easily testable until the problem is a big mass or you have significant symptoms. And two, a lot of these affect children at a much higher rate than adults, and children can't go, hey, I can't speak right anymore. I might have a tumor in my brain. They don't generally say things like that. So they're a lot harder to monitor for these things because they are non-communicative in that sense. What they basically did is they said, hey, we're going to take a sample of your blood and check it every time for a period of days, weeks, and months, and by months they mean like almost a year, so this is a very long study, and check to see if you have any disease after initial treatment. And these graphs are a little bit hard to read, but basically, like, this is a good example one. This is prior to treatment, so you can see some of that disease still there. Then they started treatment. This was a time point after the start of treatment, so you can see whoop, it went down, continue to monitor, continues to go down, and then we come back after from this time point to this time point is actually quite a bit of time. I think it's something like 12 months. It jumps up again and then jumps up even higher. So we were able to go, hey, they were in remission, but now it's come back. So we can actually go back and guide our patients much more quickly than we were able to do prior. We don't have to look for big markers. And the benefit of this is multifold. One of the benefits of this over our standard current methods is that it's whole genome, which means that we can look for your specific gene issue. Because cancers adapt, this makes it hard to target one specific gene. So for example, we're like, oh, it's this type of gene is almost always present in this cancer. That's true, but sometimes it'll mutate and it'll lose that gene, so you can't find it anymore, but it'll have all the other genes that it's got messed up still in place. So that's why whole genome sequence is better, because the cancer can't change its entire genome. And so if you get like a 95% match for that cancer genome, that's probably an indication that that cancer has come back. The other thing is that it is really good at very small amounts. It is capable of detecting one tumor cell in 10,000 to 100,000 normal cells. That's like finding a golf ball every single time you looked in a giant pit full of those like play pits of balls, just finding one golf ball in that pit every single time, 100% guaranteed. And that's actually not an accurate representation. That would be like a play pin the size of a swimming pool filled with those play balls. That's what you're looking at. So it's actually a very impressive rate. So the reason and that's the whole reason why we do this. And it is actually extra beneficial because now we're doing whole genome sequencing to look for this. We can go back and look at those patients that do show up positive, and because we now have their whole genome sequenced again, we know how to treat it better and target it better because we can look at the mutations that have occurred and go, hey, it's actually changed in this way. This treatment is no longer effective. We're going to switch to this treatment now that it's recurring again, and we will be much more effective because we're not trying something that doesn't work because we already know that it won't work. It's actually very important. Why do I bring this all up? And what should you take away from this? One, we are using whole genome sequencing to test for very tiny amounts of residual tumor popping up in cancer patients, and it is very effective at finding it. The reason why it's important is whole genome sequencing will be a standard of care for cancer and other diseases probably in the future. I would say probably in the next 20 years, whole genome sequencing will be the default thing they do. If you come in and they're like, okay, you've got cancer, first thing we're going to do is we're going to take a sample of it to test to see if it's cancer, and we're going to do a whole genome sequence of it. That will be the first steps that they do before they do literally anything else. They'll go, oh, you might have a tumor, we're going to take a sample, we're going to run it through the normal tests, and we're going to do a whole genome sequence of it. So there you go. All right, does anybody have any questions on anything I covered there? Before I let you guys go, I know you guys are itching to go. What makes you guys feel any better, I forgot and juked the last class and was like, oh, wait, come back. I want to talk about the lab practical so that you guys have a chance to kind of be prepared for it as it's coming up rapidly. And I know you guys have lots of other exams, such as the one you guys just took. We're going to talk about this real quick. Lab practical. It is very similar to the last one, but it is not cumulative in the sense that I'm not going to ask you guys random stuff from the past. It's going to be cumulative in the sense that I might ask you tests that we learned in the first half of the semester that were still relevant for this semester. For example, like MSA plates. We used that in the unknown identification in the last half of this semester. That's fair game. Gram staining, you have to be able to rate gram stain because that was part of the unknown identification. But I'm not going to be like, hey, who invented gram staining? In what year? And that's Christian Graham in 1884 or 1844. No, 1884. See, I don't even know. There you go. I would not pass that test. Okay, so I'm not going to ask you random obscure stuff, but I'm going to ask you stuff that's relevant to what we did in the last half of the semester. It'll be in the same kind of format with about 70 questions with 23 to 24 stations where you guys can kind of move through the stations. There will be no skills check. There will be a big bonus section, which is 5 to 7 questions based off of the cool science thing of the week. And that does actually impact people's grades. I mean, there are some people that were like, I am going to fail this, and then pulled out of failing range because they got bonus points. So keep that in mind. It's a good idea to attempt them. They are somewhat harder, but I don't make them impossible. So keep that in mind. Sorry. And this part is wrong. This was from last semester. You guys will have it on Tuesday. You guys will have it on Wednesday, Thursday for you guys. So Thursday is when you guys will have your lab practical. So if I were to go over this real quickly, I'm going to give you guys a couple of example questions that you might see on it, just so you can get a feel for it, right? I might go, hey, PCR, just learned about this. What's the first step of PCR? Okay, we just talked about this. Come on, guys. What's the first step of PCR? Denaturation. Very good. So denaturation stage. We heat everything up really, really high. What temperature do we heat it up to? 95. Very good. All right, awesome. So 95, very, very close to boiling. So after we heat it up, and that separates the template DNA out, we want to cool it down just a little bit. What do we call this secondary stage? We're letting the primers anneal. Very good. So it's the annealing stage. And this one's a lot cooler. What temperature do we bring it down to? There's actually a pretty broad range here. So yeah, about 50 to 65. I think in here it says 50, 56. It's actually dependent on what your primers are, and it tells you what temperature you make. So it's a very broad range. And lastly, to turn on the Taq polymerase, we have one last stage. That last stage is called? What was that? Elongation. Very good. Elongation or extension, both are correct. And it is 72 degrees Celsius. So it's kind of a little bit hotter. It's not super, super cold or super hot. And hold on one second. If I were to show you guys a picture and be like, hey, what is this in the context of identifying an unknown? Which bacteria is that? What kind of bacteria is that? You would say it's gram-negative streptobacilli. Yes. So gram-negative streptobacilli. And this would be a good indicator of what steps you take next. So if it's gram-negative streptobacilli, you would then do a series of tests, and you should know what those tests are off the top of your head. You also have to be able to interpret those tests. So for example, if I popped over here, I was like, hey, oh goodness, which one of these is DNase positive? So it's able to break down DNase. Is it A or B? A? Who says, okay, let's do a quick check. Who says A? Who says B? Who didn't vote? Okay, you guys gotta vote. Try it again. One more time. Who says A? Who says B? Okay, it is, oops, A. Very good. All right, and I will actually put up, I'm gonna let you guys go in here in just a second. I will put up a study guide and a fill in the blank thing, but it is good to start going over these things, really making sure that you understand how to analyze each of these tests. You kind of like know these parts of things that we've covered this last semester off the top of your head. There will be parts that are going to be identification based. We will give you a table and a chart to be able to do that, but you will have to know how to interpret all the tests on your own, okay? Does anybody have any questions? We'll talk more about this next week as well. Yes, last week of labs, yes. Week 15. And if you guys have any questions, come up and chat with me and we'll talk stuff over.